Welcome to the Bible and Science. This is Dave Prentice. I'm a retired public high school teacher, certified in all branches of science and math. I've also taught on the Bible and Science for many years, both in the U.S. and on many overseas mission trips. This is episode 31 of the Bible and Science. We've previously touched on some things about how cells work and how DNA works. Well, we're going to get into a bit more detail on that today. All known life is composed of cells. If we ever find something that's not based on cells, it's going to be front page news in all of the medical and science journals. So a great many organisms, bacteria, amoebas, and so forth, are composed of a single cell. In fact, there's probably more single-celled organisms than multi-celled organisms because there's so many of them. A little bitty bit of saliva could contain a great many bacteria, for instance. But there's also a great many types of multicellular life. And in some cases, such as elephants, those can be made of trillions of cells that work together, and they have specialized functions. Your heart does one thing, your lungs do something else, your skin does something else, and so forth. So whether it's a single-celled organism or many cells, every known living thing depends upon DNA, whether it's in one cell or it's in trillions of cells. DNA contains the instructions to reproduce every single part of the cell, including itself, the DNA itself. So remember, I've touched on this before. DNA contains the instruction for putting together every part of the cell, including itself. And then the process of reproduction is pretty intricate, so I want to go into a little bit of that. So for right now, I want to ignore how crucial parts of the cell, such as the thousands of types of proteins and enzymes, the ribosomes, the hormones, the endoplasmic reticulum, the cell membranes, the transport mechanisms, and so forth, all of those are key parts of the cell. But right now, I just want to focus on how new DNA is copied from previously existing DNA. So this is going to focus on one cell at a time, but in multi-celled organisms, this process has to happen simultaneously in many cells, because each cell, in a way, functions independently, but they also are interrelated. So you can see, if you look at some of the videos I'm going to refer you to, there's some that have likened the pace of cell reproduction to the speed of a jet engine. So I'm going to give you some links in the comments section of the podcast page, thebibleandscience.net. I don't hold the copyright on these, so I'm going to link to them, and there's some really well done videos out there to show you how this happens. So for instance, if you go to thebibleandscience.net, you're going to see that there's a link to HHMI Biointeractive. They show a beautiful video illustrating a simplified model, very simplified, of how a single DNA double helix is separated so that the two strands can be produced from one. But notice this process is a whole lot faster than what's going to be shown on any computer simulation. It's a whole lot more complicated, but the video will show you one thing at a time. So you go to that website, go to my comments page, and you can find a link to that website. The video you saw, if you went to YouTube, shows you just a little bit of what's going on, and it only focuses on one particular area where the enzymes are breaking apart things and so forth. Well, I also want to show you some simplified details of what's going on there in a diagram. There are many enzymes, for instance, starting with ligases. They split the DNA, and then they unwind it since it's a helix, they're helicases. They unwind it into two halves. One of them is going to be copied forward. The other one has to be copied backward. So you have the leading strand, which is reproduced directly, but the lagging strand going the other way, it's cut into segments by topoisomerases into short segments, which are known as Okazaki fragments. So it doesn't become tangled despite the fact that it's so long. Remember, this can be anywhere from millions of base pairs, millions of nucleotides, up to hundreds of billions. So you've got to cut it into segments so that those hundreds of billions won't get all tangled up. So then, once the new DNA strand is completed, they have to be assembled in the reverse direction. So that image is from a lady I've never met, but she's done some wonderful work that you can find on Wikimedia. That's an illustration of how DNA replicates. It starts out splitting apart, and then it has to unwind, and then it begins to reproduce each of the fragments. In one direction, they go straight ahead. In the other direction, they're cut into small fragments in order to be reproduced. And remember, this is supposed to be the result of lifeless chemicals. So in that process, what happens when a copying error occurs? Well, in the previous slide, you saw that the halves of the double helix are unlinked by ligases, and then they're unwound by helicases, and then topoisomerases cut one of the two strands into short segments to prevent it from becoming tangled. And meanwhile, the cell is manufacturing large numbers of the nucleotides A, C, G, and T, adenine, cytosine, guanine, and thymine, so they'll be ready for use. Then G and C each have 3 sites that are called hydrogen bonding sites. They have opposite charges. One side is negative, the other side is positive. So G and C match up together because they have 3 hydrogen bonding sites. A and T have 2. Usually that makes them match correctly. But if there is a copying mistake, a mutation, a mismatch, then there are further enzymes that come along, proofreading exonucleases. They snip out the incorrect nucleotide and they correct it, they replace it with the corrected one. And then DNA polymerases perform more proofreading, and they repair any mismatches that have still somehow slipped through. So if you're an atheist, you may believe that this is an amazing series of millions of mutations. I have a hard time with that. To me, this points much more strongly toward design. And then after the initial assembly is done, it's possible that there could still be some mismatches. So once again, on Wikimedia, Lady of Hats has put a wonderful illustration. If there's a mismatch, then there are going to be multiple enzymes that come along, find the error, snip it out, and manufacture and insert a new corrected segment of the DNA strand. Now, what kind of machine is operating this? A lifeless machine. These are lifeless chemicals, but yet somehow they do exactly what they need to do. So if there's an error, it gets snipped out and it gets replaced by a corrected segment. All of this is done by lifeless chemicals. So what happens if a copying error does slip through, a mutation? DNA is composed of anywhere from millions of nucleotides in things like bacteria to billions in some types of fish, mammals, and flowers. Can you believe that? Some of the things that have the most DNA are flowers. For instance, the lilies. Jesus said, "Consider the lilies of the field." Well, he knew what's in the DNA because lilies have some of the largest quantities of nucleotides in their DNA out of anything. So, okay, the DNA that's there, it contains the instruction for how to perform regulatory functions. It switches genes on and off. It contains the instructions necessary to copy itself. It also contains the instructions to assemble other chemicals into the components of cells, such as proteins. These are responsible for most of the cell's structure. So the DNA has the coding information to do the rest of the cell, but the DNA itself does not compose most of the cell by volume. So if there's a mutation, a copying mistake that slips through, it damages DNA. So if there's a mutation in one of the non-essential alleles of a gene, the variations, eye color, hair color, and so forth, then the components coded for by that segment of DNA may not suffer lethal damage. They may possibly have a neutral effect, the hair color, the eye color, something like that. But if a mutation occurs in one of the crucial parts of the gene, of the DNA, the homeotic genes, it's usually going to be harmful. Sometimes, pretty often, it'll be a lethal mutation. So remember, according to evolution, the first living things were extremely simple. There was no kind of mechanism like there is in DNA nowadays, so how did they copy it? We don't know. It's just some sort of an accident. So because DNA had not yet evolved, there must have been some far simpler, unknown mechanism used to store information and then translate it to copy the rest of the cell. So at the very beginning, there would not have been any sort of error correcting mechanisms at all. We just have some molecules composed of CHNOPs, carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur, and they wouldn't have had any sort of error correcting mechanism. So where did it come from? Well, if you're an atheist, you believe that the elaborate error correcting mechanisms were later added to DNA one mutation at a time. Now remember, there are some living things that have hundreds of billions of nucleotides on their DNA, but you have to believe that the error correcting mechanism was added one mutation at a time. And then there's an intricate system of enzymes that I touched on that prevent errors. That in itself is the result of thousands of perfectly coordinated errors. This is according to atheists. So errors produced the error correcting mechanism. Now, if you're a theistic evolutionist, then it should be pretty plain. I'm a Bible-believing creationist. If you're a theistic evolutionist, you would believe that God must have added the error-correcting mechanism much later. He created the first living cells and then decided to insert an error-correcting mechanism. But in order to have evolution, he really, really, really wanted to have evolution, so he must have overridden the error-correcting mechanism that he put into the cells millions or probably hundreds of millions of times, just so that he could get things to evolve. While the alternative would be he designed it the way it is today. The error-correcting mechanisms in DNA don't encourage evolution, they prevent it, not enable it. So the rest of the cell is composed of many, many, many other parts, and I'm going to give you a brief summary. There's another great video on how DNA is translated into proteins. The nucleotides A, C, G, and T, which have nothing to do, or they are not part of the proteins of a cell, but they're translated into proteins, which are the structural components of the cell. It's like the blueprints of the cell. So the video that you see there talks about the cells doing something in the nucleus. Not all cells have a nucleus, but still the function is the same. The process is basically the same, whether it's a prokaryotic cell without a nucleus or a eukaryotic cell that has a nucleus. So here's a brief summary of what happens, and then you can go to that video and look at it. The cell sends a chemical message, for instance, a hormone, to the DNA. The DNA is down in the center of the cell, and it indicates, the messenger indicates which protein is needed. So there are many, many, many enzymes Certain enzymes go along and they locate a start code in the DNA segment that codes for the protein that's needed. And then the ligases and helicases, they don't unzip the whole DNA strand, just the segment that's needed. They unzip the two halves of the DNA strand at that location, and there's also a stop code at the end saying, okay, that's it for this protein. So in a process, it's called transcription. Other enzymes, such as RNA polymerases, they use one of the strands to put together messenger RNA. Messenger RNA will be used to line up the amino acids that make up the protein. And I have referred repeatedly to A, C, G, and T. Well, in messenger RNA, instead of thymine, it uses uracil. Okay, this is the genetic code that scientists have discovered. There are many genes in DNA that contain the instruction to manufacture proteins. There's other parts that don't. There are regulatory functions and so forth. But the ones manufacturing proteins, the coding genes, they made out of hundreds of thousands of the nucleotides A, C, G, and T, and the messenger RNA comes along, matches up with those. So the coding genes, the ones that are going to produce the proteins, they're broken down into segments of 3 called triplets. Each triplet contains the information to put one specific amino acid in a protein as shown there. And when I talked about the origin of life, there are many, many, many different types of proteins, different types of amino acids that can form, but only 20 very specific ones are used in DNA. So each triplet contains the information to put a specific amino acid. For instance, you'll see TGG, thymine, guanine gives you tryptophan, and all of the other ones, oh, wait a minute, when you get down to the third one, there are 2 different sequences, TTT or TTC, those both produce phenylalanine. And when you go down to some of the ones near the end of the list, you have GTT, GTC, GTA, GTG. These are 4 different sequences that will also put a valine into the forming DNA. What? That sounds awfully inefficient. Remember, though, as I've pointed out before, that the DNA is copying in both directions at the same time. In one direction, it's going to read, for instance, ACT, but in the other direction, it may be reading ACA. The DNA is made up of millions and millions of little triplets, and they can be read in either direction. So the fact that they can be read in either direction allows for the same amino acids to be coded by more than one structure, because they can be read in both directions. Now remember, this is supposed to be an accident of evolution if you believe in evolution yourself, or I would look at that and say, wow, that's better computer programming than any human being has ever come up with. So this is how cells manufacture proteins. And again, you can go to the comments section to get a link that takes you to the animation. So once a segment of messenger RNA is complete, it's transported by other components of the cell. There's so many components of the cell, to an area of the cell known as the endoplasmic reticulum. This is where the actual manufacturing, the translation from genetic information into proteins is done. So the mRNA attaches to ribosomes to prepare for the manufacturing to begin with. And then, instead of messenger RNA, now we have transfer RNA, tRNA, which is also called a codon. Each tRNA includes 3 of the 4 possible nucleotides, A, C, G, and U instead of T. So they move into place and they match up with 3 nucleotides on the messenger RNA. So each transfer RNA molecule matches with exactly 1 of the 20 amino acids used in living things. If you go back to the genetic code on the previous page, I didn't mention on there that these are all left-handed amino acids with the exception of one that only occurs in one form. But they're all left-handed. Right-handed amino acids exist in nature, but they're just not used in living thing. The transfer RNA is going to make sure it gets the right thing, the right part to put in it. And again, this is from the Wikimedia user, Lady of Hats. Fantastic stuff she does. Now we have our DNA in place. Well, it's necessary for the cell, but it's not sufficient for the cell because there are a whole bunch of other parts besides just the DNA. It would be like if you put together building plans to manufacture a new house. Okay, that's great. I've got the plans. Now you need to do something to put them together. You need to go out and get the lumber and the concrete and the nails and all of those things. So here's the DNA. It matches up so that we get messenger RNA and then we get transfer RNA. So the transfer RNA has attached itself in the ribosomal area. So as each transfer RNA molecule puts its attached amino acid in place, the chain grows, the chain of amino acids. So then the transfer RNA releases that amino acid, which fastens onto another one, and then it returns to it, being the transfer RNA, returns to its former location where it's available to be recycled over and over and over again. Proteins don't consist of just one or two things. They consist of hundreds of amino acids, but they're in a precise sequence. And even if we put them in a precise sequence, that's not enough. A long chain of amino acids is useless. Before it becomes a functional protein, it has to be folded into a correct three-dimensional shape. This is done by previously existing proteins needed to make more proteins. They must already be present in order for the new proteins to assume their correct shapes. So there's a link in there to a YouTube video that shows this very nicely. The proteins are folded by chaperonins. It sounds cute, right? Somebody said, okay, let's have a chaperone. So they're folded by chaperonin complexes, as seen in the lower right of the page. And when the chain of amino acids goes in, it's folded to the proper shape, and then it comes out as a properly folded protein molecule, as you see on the left side. So it's still not sufficient. It's got to be transported to the rest of the cell where it's needed. And there's a whole separate mechanism for that. There are what you could call, I guess, roadways, microtubules in the cell. And so those move the components along to where they've got to go. And if you look at the little cartoonish thing at the upper right, all it shows is a circle, but it's a precise shape. This is called a kinesin. It's sort of like the trucking network of the cell, and it's got to go along the microtubules, which are like the roadway. Gee, can we see any sort of a thing here that might be beyond what random chance can do, you think? I think so. So what happens if something goes wrong and a mutation changes the DNA? Well, if you read the comic books like the X-Men or something, they're very entertaining, but they're not realistic. Mutations don't produce super beings like the X-Men. If they did, and you believe in evolution, you ought to try to go get a house that's right next to a nuclear power plant so you could be exposed to radiation and become a mutant and gain superpowers. No, we all recognize that doesn't happen. Mutations don't add any genetic information to the cell. All they do is take previously existing information. Where did that come from? It was already there. In most cases, changing the previous information causes damage. So then, if the effective segment of the DNA that contains the coding instructions to produce a non-essential feature like hair color or something, well, there may not be obvious harm to the organism. But if the instructions for an essential feature, such as blood cells, hearts, lungs, brains, and so forth, if that part of the DNA is damaged during reproduction, then the organism will not function normally, and it may even die young of cancer or of other deadly conditions. There's so much more to cover. People have made careers out of studying the operation of living things and genetics and so forth. So this is just intended to barely scratch the surface for you. We'll continue on in the next session, and this is sort of pointed toward the idea of design in nature. And as we go along, we'll see a great many more things that indicate that life is not random, it's not the result of chemical processes, but it is designed. So, see you on the next slide.